But what we're going to do today is we are going to talk about the sequencing experiments, or we're going to go through the actual protocol that we would have followed to do the experiments. And then I have a supplemental handout, again, just going over the key points for what we did, and then to talk about sequencing. At the end, I'll just mention the assignment that I posted yesterday. And so we'll talk about that, but we'll talk mostly about that on Wednesday, because really we don't need all this time to do the sequencing in bioinformatics. It's just that in lab, we would have needed that lab time to do all these experimental procedures. But that's how the syllabus is, so I'm just going to follow what's there. So I am going to be sharing my screen, so that again means you need to talk to me since I can't see chats. So we'll start by looking at the regular lab protocol that we would have followed. So this is the protocol for the identification of unknown number 3 by PCR and sequence analysis of the 16S ribosomal DNA. So can we all see this handout?
Yes.
Yeah.
So this is part of unknown number 3, which was just learning how to use certain databases to identify unknown organisms by using a sequence of DNA. So we're going to talk about how we actually would have extracted the DNA and then used it for sequencing. So what we would have done in the first lab is actually prepare our DNA template from bacteria. So each group would have received 2 unknown organisms to play with. Then we would have set up a PCR reaction to amplify a piece of the ribosomal RNA gene, approximately 779 base pairs long. And then on the second day, we would have analyzed your PCR product using agarose gel electrophoresis to see if, in fact, you got a piece of DNA approximately 800 base pairs long. And then on the third day, we would have used that sequence because we would have gotten the sequence information back, and would have used that sequence to identify your unknown organism. So we are using a piece of the ribosomal RNA gene. And the reason why we use the ribosomal RNA gene is because you all know that ribosomes are the little machines, right, for translating the messenger RNA to make polypeptides and proteins. And so all living organisms have ribosomes. And ribosomal RNA is a very important structural component of the ribosomes. So all living things have ribosomal RNA genes that when they are transcribed into RNA, that is the ribosomal RNA used, right, to help build the ribosomes. So this is the actual piece that we're going to amplify. It's just sort of FYI so you can see it. The thing about the ribosomal RNA gene is that there are certain regions of the gene that are highly conserved, and then there are other regions that are variable. They are different for each organism. It's that difference that we can use to identify, right, various organisms. So that's why we use the ribosomal RNA gene. So on the first day, which would have been today, each student or each group, and you would have worked in groups of 3 students, would have received 2 unknown bacteria. And so you would have put those bacteria into 2 separate microfuge tubes. So you're probably familiar with those little tubes we use for microcentrifuges. And then you would have boiled those bacteria for 10 minutes. And the idea behind boiling is to rupture the cells and release the chromosome, right, to release the DNA. But after boiling, you would need to centrifuge it in a high-speed centrifuge. So not the little centrifuges that you may have used before, but a very high-speed centrifuge. And the reason why is we need to separate all the junk, all the cellular debris from the DNA. And so when you spin down these tubes at very high speeds, you form a pellet in the bottom, which is all the cellular junk. In the liquid, which is called the supernatant, that's where the DNA is floating around. And so we then use that supernatant as our source of DNA. So you boil the bacteria, rupture the cells, centrifuge them at high speeds in order to separate the DNA from all the cellular debris. Once you've done that, you would start setting up your PCR reaction. And so again, we have these little plastic tubes. They're actually in strips. And there'd be 3 tubes in your strip. Your unknown organisms be labeled A and B just for the sake of something easy. So organism A would go into the A tube and organism B in the B tube, and C was a control. So to set up your PCR reaction, remember there are 4 key ingredients you need for PCR. You need template DNA, which you just made by boiling and centrifuging your bacteria. You need a buffer, right, because all reactions are done in a buffer. You need the DNTPs. Does anybody know what the DNTPs are? DNTPs? Those are the ATGCs, all the nucleotides. And then we have polymerase. What is special about the polymerase used for PCR? What was special about the PCR or about the polymerase used for PCR? So what's special about the polymerase used for PCR? We talked about this.
What's your question again?
What was special about the polymerase used for PCR? Remember, it's an enzyme. And what was special about it?
Did it have to be used from like a special bacteria that could be heat resistant?
Yep, so remember it came from a thermophile. So it's very heat tolerant. That's very important. So you have your PCR mix, which included the buffer, all the nucleotides, and the polymerase. And then you have some sterile water and your forward and reverse primers, right? So remember, we need primers. And then you would add that reaction mixture to your tubes A, B, and C. And then you add some of your DNA from tube A into your PCR tube A, from tube B into PCR tube B, and then tube C was just a control, so you added some water to that. So your tubes A and B have your DNA in there, and then we put those tubes into an instrument called a thermocycler. And the thermocycler just heats, cools, heats, cools, heats, cools over and over and over again, because we have to heat up our samples to 95 degrees Celsius. That's 5 degrees below boiling. And the reason why is we have to separate our double-stranded DNA into single-stranded DNA. So we've got to heat up to 95 degrees Celsius. By using DNA polymerase from a thermophile, that enzyme does not denature, right? Because 95 degrees, that's extremely hot. Then we cool it down to 58 degrees Celsius. And the reason why is we need the primers to bind to the DNA. And so the primers are specific. They will only bind to the region of DNA that we want to amplify. And the binding is base pairing. And so we cool the sample down to 58 degrees Celsius. It allows the primers to kind of wander around, touching the DNA. And then when they find the complementary bases, they can pair with that and stick to the DNA. But we need to have the temperature warm enough to allow that to happen, but cool enough so that we don't separate the primers from the DNA. Because if you heat the DNA too much, you'll separate the primers. And we need the primers, right? Because we need that region that's double-stranded for DNA polymerase to hop on and start replicating. So after we give time for the primers to bind, we call that annealing. Then we raise the temperature back up to 72 degrees Celsius. And the reason why we have to do that is our DNA polymerase is thermophilic. It needs heat in order to function, but not too hot. If it's too hot, then the primers will separate from the DNA. So we can't make it too hot, but it's got to be hot enough so the polymerase will function. So we raise it up to 72 degrees Celsius. RNA polymerase hops on and starts to replicate. And then we repeat the process over and over again. We heat the DNA back up to 95 degrees Celsius and cool it back down, allow the primers to bind, heat it up a little so polymerase can function, and then repeat this over and over and over again. And in this lab, we do that 30 times. So it takes the thermocycler about 2 hours to do 30 cycles. So we would put the tubes in and start the machine. And then by then, the lab was pretty much over. So you would not get the results of your PCR until the next class. But we extract the DNA by boiling. We set up a PCR reaction, hopefully to amplify our piece of DNA that's approximately 800 base pairs long. The way we verify that, in the next class period, we would run an agarose gel. And so you should all be familiar with agarose gel electrophoresis. In our class or here at Rockville, we use fast gels. And so the fast gel allows us to see results in as little as 5 minutes. And so the agarose gel is encased in a plastic cartridge. And the buffer is included in the gel. So the wells are basically dry wells. So it's not like this diagram shows. There's no buffer floating on top of it. The buffer is incorporated into the gel. And the plastic cartridge, the plastic case, has little holes in it where the wells are. And so, of course, we add our loading dye to the sample to put it into the well. It just helps us to see that the sample is there by making it blue. And the only concern here is that when you put the tip in the well, you want to be careful not to poke through the gel and squirt your DNA in between the gel and the plastic case. So you just put the tip in the well and release your sample gently. And so once all of our samples were loaded, we would run them and they would separate in 5 minutes. Now, as you recall, since we're estimating the size of our fragment, we have to use a DNA marker. And the DNA marker is a series of fragments of known size. So we know how big all those fragments are. And so we can compare our experimental bands with the DNA ladder. And so what we hope to see is that each group got one band that corresponded to the 800 base pair fragment. So if you got that band, that would mean that you amplified the correct piece of DNA and got the right size piece. Now, ideally, each group should have a band for each of their unknowns. In practice, we don't get that. I think the best that I've ever gotten are 10 or 11 bands total. So each group got a band for each unknown for most of the groups. But it does depend on your technique. And in this experiment, micropipetting is critical because you're pipetting samples as small as 1 microliter. So if you're not careful, you may not be pipetting anything at all, or you may pipette way too much, which screws up the reaction. So again, paying attention to detail is important. So I've never had a case where every single group has gotten both of their unknowns. Almost every single group has. And sometimes there's a group that doesn't get anything for either unknown. So technique does matter. But hopefully you would have verified that you had at least one band, so you had at least one unknown that you could get sequence from and use that sequence to identify it. So if you verified that you actually had a sample with a PCR product that worked, then your sample would get sequenced. And we are going to learn about the sequencing process soon. But what we do, there's actually a nearby lab right nearby to the Rockville campus. I forget the name of the lab, but all they basically do is sequencing. All day long they sequence. So you can drop off your samples on your way home from work, and the next day they email you your sequences, right? So they run them, and the computer scans them, and then they email you the sequence information. So it truly is incredibly fast these days and relatively cheap. I think the last time that I heard, they only charge $5 per sequence, so that's pretty cheap, and especially given that it's so fast. So in the last lab period, we would have those sequences so we could then analyze them. And so there were two ways to do that. One was to use the ribosomal database. And when I tried to access this this morning, the site was not available. So I will try it today with you to see if it works. And then the other site is the NCBI site, and we are going to talk more about that later. That's part of your assignment that you'll be doing later. But you have the sequence for your unknown, so you need a way to match that sequence to known sequences of organisms. So there are different databases, like the ribosomal database, where you can input your sequence by copying and pasting to make it easy. And then these different databases, the software they have actually aligns. It matches your sequence with known sequences in the database. And the greater the alignment, the better the match. And so you can use this to identify unknown organisms. Or maybe if you have an unknown gene, you don't know what that gene does, you can input the sequence and see if it matches a known gene. Maybe the same thing, you have an unknown protein, but you have the amino acid sequence. You can use that sequence to identify your protein and its function. So these databases are extremely important and useful today. And so scientists from all over the world can input their sequences. And so these are free services, right, that are provided to scientists anywhere. But the benefit is that you can sequence a gene, a protein, and then use that sequence to identify what that gene or protein is. And you can use it to identify unknown organisms. The process is becoming so easy that there are actually some hospitals that sequence unknown bacterial pathogens causing an infection in a patient so they can determine the best treatment to use. So a patient can come in with this infection, and in the hospital they can actually isolate and sequence the ribosomal RNA gene and use that sequence to identify that organism in a day. So they can do this very, very quickly, and that can help determine the best antibiotic to use to treat that pathogen. So some hospitals are able to do this. So, normally it would require, more time in order to identify the organism and determine its antibiotic resistance, right? So you could find the best antibiotic to use. But there are some hospitals that can do this all in a day. So that's really good for the patients because they can get treated much more quickly. So let's see if the ribosomal database will open up. Oh, and of course it's working now. So let me open up a new tab for this here. And this morning it absolutely would not work. And then boom, now it's working. Come on. Well, it was working. I'm not getting anything. Okay, now I get it. Yeah, so we're here, right? So I have some sequences to play with, so I'll just go grab one of these sequences. So we have instructions, right? Select the SEQ match. So that's right here. And we're going to paste our sequence into the window. And we want to change source to isolates and size to both. Source to isolates, size to both, and then submit. And so how long this takes for, any of the databases depends on how big your sequence is and how busy the web traffic is to this site. So we got an almost instant result here. So it gives us our domain, phylum, class, order, family, and then we can view the matches. And we'll see our sequence matches Enterobacter aerogenes, right? So usually the first match is the best match. And notice here it says genus Klebsiella. So let me actually show you the other database, which I'll talk more about on Wednesday, but the easiest way to get there is just to type BLAST, and then you get here. And so BLAST is the basic local alignment search tool. So this is the database produced by the NIH. And so we're going to do a type BLAST. Again, we paste our sequence into the window. And the only thing that we have to change here is to show results in a new window. So normally that's not checked, but since I did this morning, it remembers, and then hit BLAST. And this database is actually used by a lot more scientists than the ribosomal database is. So this one is always busier and takes more time. So this can take a few minutes, again, depending on web traffic or how many people are using this database. But this does give us some more information. It's a more complex and comprehensive database. But I just want to show you something that will come up in the results. So it could take a minute, it could take a few minutes. It just depends. Okay, here we are. So notice that our query length was 777. That's the number of nucleotides in the sequence, right, that I pasted into the window. And the descriptions, again, our first match is Klebsiella aerogenes. So here, oops, Enterobacter aerogenes, but the genus is Klebsiella. So why the discrepancy? If you scroll down and look at more matches, you'll see at some point Enterobacter aerogenes. That's actually the old name. It's a name that we still use in lab, but this organism has been reclassified into the genus Klebsiella. So Enterobacter aerogenes is actually now Klebsiella aerogenes. Not all scientists have switched over yet, so you'll still see Enterobacter used. But the organism that matches is the Klebsiella / Enterobacter aerogenes. And notice it used the 16S ribosomal RNA gene, right, to do the match. But we have two techniques, right, to identify our organisms. This one doesn't give you quite as much information as the other database does, but it does give us matches. And down here, we notice the new name being used, Klebsiella. But these are two databases that can be used to identify your unknown using the sequence, right, that you'll get. So, but this did not work this morning. It just would not connect to the website. So let's go back. Right, so we've seen the two ways, once you get a sequence that you can use to identify your unknown organisms. So either the ribosomal database or blasting using the NCBI database. So I prefer the NCBI database because there's a lot more data there that you can use. And the assignment that we'll talk about at the end uses the NCBI database. But it's pretty easy to do. So, any questions so far on what we've talked about? So we'll look at the other handout, a handout that I created back in the spring, again, to sort of what we just talked about, but also, oh yeah, question? No question? So also to talk about how the sequencing reactions work. But first, for review, we just went over the main handout. And again, to summarize, we extract DNA from unknown bacteria by boiling them. Then we set up the PCR reaction to selectively amplify a 779 base pair fragment. It's approximately that. You're not going to get that exactly when you do the actual experiment and the sequencing, but it's around that number. And we're using the 16S ribosomal RNA gene. We then would run an agar-Roche gel to verify that the DNA fragment corresponding to approximately 800 base pairs was amplified. And then you obtain a sequence from that amplified DNA fragment and use it to identify the unknown bacteria. And again, the ribosomal RNA gene is commonly used because it's found in all living cells. And as far as the size goes, it's not too big. It can be easily sequenced, but it's big enough so we can see differences between different organisms. PCR requires 4 ingredients. We need our template DNA to be amplified. We need that special DNA polymerase, which is thermostable, so it does not denature. We need all the nucleotides, which are called dNTPs for short, so the ATGCs, and we need primers. And the purpose of the primers is, one, to provide a place that's double-stranded so DNA polymerase can hop on, right? Because DNA polymerase needs a region that's double-stranded before it can hop onto the DNA and start to replicate. So that's what the primers do. The primers are also specific to the gene of interest, to the region of the DNA that we want to amplify. So the primers are made specifically to base pair to the gene of interest, because that's the place where DNA polymerase will hop on and start to replicate. So only the gene that we want to amplify will get amplified. So again, to go through the cycles, heat up the DNA to 95 degrees Celsius to separate the 2 strands. Then we cool it down to 58 degrees Celsius to allow the primers to bind to the specific region that we want to amplify. Heat the mixture back up to 72 degrees Celsius so that the RNA polymerase can function. It needs some heat to function and replicate, but not too hot, right? We do not want to separate the primers from the DNA. And then after the polymerase runs for a certain period of time, we repeat. Heat it back up to 95 degrees, cool it down to 58, heat it back up to 72, and over again, 30, 40, 50 times. In this experiment, we only did it 30 times, but often they do it up to 50 times. And that way you can dramatically amplify the sequence of interest, so you can increase the amount of DNA, because remember, the DNA doubles with every cycle. So the fancy name for the sequence of interest is called the amplicon, right? So the DNA that you amplified is called the amplicon. And so the amplified DNA is then run on our agar-Roche gels to verify that we amplified the correct size piece. If you got a band in the gel, that means you had a PCR product that can be sequenced. And so then we would send that sequence out and get it back, and then use that sequence to identify the unknown, either using the ribosomal database or using the BLAST NCBI site. So how does sequencing work? Well, the most common technique for sequencing is the Sanger dideoxy technique. So the Sanger dideoxy chain termination method. It was created several decades ago. It won a Nobel Prize. It was not the only technique that was being developed at the time, but it is the best one. and it is the one that is used almost exclusively today. And we will watch a little video on the technique later. But Sanger dideoxy sequencing uses the exact same 4 ingredients that PCR uses. We have template DNA, we have our thermostable DNA polymerase, we have all the nucleotides, and we have primers. In addition, there's a 5th ingredient. And the 5th ingredient are dideoxynucleotides. So a regular nucleotide is a deoxynucleotide because DNA is deoxyribonucleic acid. But these are dideoxynucleotides. And what's different about them is that they do not have that hydroxyl group, the OH group, needed for nucleotides to be joined together. So there is no OH group on a dideoxynucleotide. So that means if DNA polymerase incorporates a dideoxynucleotide, that's it. Elongation terminates. No more nucleotides can be added after. So once the dideoxynucleotide is incorporated, no more nucleotides can be added after that. So elongation terminates, and you end up with a shorter piece, a smaller fragment of DNA. So you have to set up 4 separate reaction mixtures. All 4 reaction mixtures have the first 4 ingredients: our templates, our primers, our regular deoxynucleotides, and of course DNA polymerase. And then each reaction mixture gets 1 of the dideoxynucleotides. So we got 4 tubes, 4 reaction mixtures, each one with a different dideoxynucleotide, right? Dideoxy-A, dideoxy-T, dideoxy-G, dideoxy-C. So 4 different reaction mixtures. And so what's happening in those reaction mixtures is that the DNA is being replicated. But in the process of replication, most of the time, a regular dideoxynucleotide will get incorporated. And so replication will proceed. But every once in a while, a dideoxynucleotide gets incorporated. And so replication stops. And so because you have a random mixture, sometimes as the DNA is replicating, it may replicate, the entire fragment and not incorporate any dideoxynucleotides. But at other times, it will incorporate dideoxynucleotides at different locations. And so the end result is that you generate fragments of all different sizes. Each fragment terminates with a dideoxynucleotide. So again, it's random, right? The incorporation of the dideoxy is random, but there's enough in the reaction mixture so that every possible size fragment will be generated during replication. So some of the fragments are short, right? Because they incorporated a dideoxynucleotide right away. Other fragments are long. It took a long time before a dideoxy got incorporated, and there's everything in between. So every possible size fragment gets generated. Each fragment ends with or terminates with a dideoxynucleotide. So we have our 4 reaction mixtures. Each one has a different dideoxynucleotide. After the reaction has allowed to happen, each reaction mixture, each tube, has a bunch of different size fragments in it. Each fragment terminating with whatever dideoxynucleotide was put into that tube. So now we have to separate the fragments. And we are going to use electrophoresis, but we're not going to use agarose. Agarose is no good for this. We're actually going to use polyacrylamide. So polyacrylamide is far more sensitive than agarose. So what that means is a polyacrylamide gel can separate fragments that differ by one base pair. So a single base pair, and the fragments will separate in a polyacrylamide gel. Agarose can't even come close to that. But in polyacrylamide, one nucleotide difference, one base pair difference is all you need, and you can separate those fragments. So that's why we use a polyacrylamide gel. And these polyacrylamide gels are pretty large gels because you normally get many, many fragments in your reaction mixtures. So you need a large gel to separate them. So these are very big gels. So when we separate them, we get something that looks like this. And this is just an example, right, to illustrate the process. So each well, each lane was one of the reaction mixtures. In this first lane, our reaction mixture contained a dideoxyadenine. In the second lane, a dideoxythymine. Then this lane had a dideoxyguanine and a dideoxycytosine. So there were different size fragments in each of those reaction mixtures. I just am using a simple example, right, to illustrate. Normally you'd have hundreds of fragments on your gel, but we just have a small number here to illustrate the process. So we separate the fragments, right, by electrophoresis using an electric current. Just like with agarose gels, the smallest fragments move the fastest, right? They move the furthest. Larger fragments are slower. They lag behind. So small fragments move the fastest, larger fragments are slower. Each fragment terminates with a dideoxynucleotide. So we know this fragment here, it terminated with a dideoxy-T. This fragment terminated with a dideoxy-A. So we know all these fragments terminate with a dideoxy-A, all these terminate here with a dideoxy-T, all these terminate here with a dideoxy-G and a dideoxy-C. So we know that. Each fragment, it terminates with that dideoxynucleotide in that particular lane. So are we following?
Yeah, and just to clarify, you said that anytime it stops, it's random. If it gets incorporated into the DNA, that's when it stops.
Right, so in that reaction mixture, right, with whatever particular dideoxynucleotide is there, as DNA polymerase is replicating, it will grab the complementary nucleotide. If that happens to be a dideoxynucleotide, that's it. Elongation terminates, and that fragment is shorter. But we know that fragment ends with that particular dideoxynucleotide. So when we separate all those fragments in our polyacrylamide gel, we know that each of those fragments ends with, it terminates with that dideoxynucleotide. And so we read our gel from bottom to top. And that's how we get the sequence. So in this example here, our first nucleotide is C, right? Because we're in this lane, and this is the smallest fragment, and it ends with a C. Our next fragment ends with a T, G, C, TT, A, G, A. And that's the actual sequence. So by reading the gel from bottom to top, you're reading the sequence of that piece of DNA. So again, C, T, G, C, TT, A, G, A. And that's the sequence. Now, if we're looking at what is the actual sequence of each fragment, we know this little fragment here ends with a C. And so that's our first fragment here, right, the C. Our next fragment we know ends with a T, but it's CT because the smallest is C, and the next one is T. And remember, we separate the fragments based on a single nucleotide. So this fragment is CT. Now this fragment ends with a G, but we know it starts with C, and then T, and then G. So that's this fragment here. Our next fragment, we know it ends with a C, but it started with a C, and then T, and then G, and finally ends with a C. So that's that size fragment here. And so on and so forth. And the last fragment is the whole thing, which is CTGCTTAGA, which is the sequence that we read from top to bottom. CTGCTTAGA. And that is our sequence for the largest fragment. So each fragment kind of builds on the next fragment. So the smallest one ends with a C, and the next one ends with a T, but we know it started with a C, right? The C came before the T. And this fragment is CTG, and then CTGC, and so on and so forth. So all these bands, these are all the fragments that correspond to these bands. In reality, there'd be hundreds, if not thousands of bands. But this illustrates the main idea that each reaction mixture has one dideoxynucleotide. So as DNA is replicating, when it incorporates a dideoxynucleotide, that's it. Right? Replication stops for that particular piece of DNA. But this is happening a lot in that reaction mixture. So all possible size fragments will be generated. Each fragment terminates with that dideoxynucleotide. We load those reactions or those reaction mixtures onto the polyacrylamide gel, which separates our fragments based on a single nucleotide. And we read the gel from bottom to top. That gives us the sequence for our piece of DNA. So questions? Well, let's watch our little video. It's a cute little video, but it does have a mistake in it, and we'll see if you guys can figure out the mistake. But
Grammarly does more than errors.
these are stupid commercials. Stop this again. So that helped to explain things?
Yeah, that was a really good video.
Yeah, it's cute, but it does have one mistake. And did anybody notice that mistake? Well, watch it again, and this time look carefully, right? Because again, attention to detail matters, right? So let's see if you can find the mistake. Because there is a mistake in this. So did we see it? Anybody see the mistake?
No, was it in the words? Was it in the text? Or in the picture?
Right, so this part here. What's wrong?
Should it be RNA primer instead of DNA polymerase?
Say that again?
I'm not sure, but should it be RNA primer instead of DNA polymerase?
No. The primer was there, and then the polymerase hopped on and started to replicate. But do we see a problem with the replication?
Oh, is it going from the wrong end?
It's going the wrong direction.
Yeah.
Right? They mislabeled their DNA. This piece should be 5' and 3', and that makes this going in the 5' to 3' direction. So they just needed to change these labels here, right? 5 prime and then 3 prime. So then this way the polymerase is going in the right direction. So a little mistake, but this was produced by students. So, but it's a cute video. Right, so their polymerase was just going in the wrong direction. Blasted. Oh, I shut down that other handout. But so any question on how we do this? Let me said I shut down that handout. Everybody like my cartoon here? So do we all understand the Sanger dideoxy sequencing, or you just need some more time to absorb and assimilate it? I mean, we're going to go over some of the stuff again on Wednesday. So let's talk about a little bit about the homework. It's not due until Monday, and I'll go over this in more detail on Wednesday, but just to give you the heads up in case some of you want to play with this now, because it's not complicated, but there is an assignment. So it's due Monday. It's only worth 12 points. And the the main handout is here, and it's a big handout, so, but it goes through how to use the NCBI database, and so there's step-by-step instructions on how to do things, and then there are some questions to answer that you'll turn in. And so, but when you go to turn them in, I actually have a separate sheet for you to use. And so that's the answer sheet. And this is what you'll actually submit, because it's just a one-page version with the questions on it. And you'll type the answers into here and then just submit this one sheet. Yeah, question? I can't see your comments, so you got to speak. Yes? No question? Right, so just the answer sheet is the one that you'll actually use, right, to submit, because it's much shorter. The original handout is 12 pages long, but 6 of those pages are just supplemental information in case you were interested. But we'll go over this in more detail on Wednesday, but you're welcome to start playing with it now if you want. As I said, it's not really hard to do. And so Wednesday, again, we'll go over the homework and talk more about this stuff again if you guys have any questions. Next Monday, it will be a review for the lab exam. So anything else?
Next Monday is our lecture test too, right?
Right, yeah. So this week is the last week for lecture material. On Wednesday, we have a short lecture on TB with a couple of videos to watch, and then that'll be it for the material. And then exam 4 will be on Monday, lab exam review Monday. Wednesday is the lab exam, and the lecture is a review for the optional final.
Is there going to be review for the fourth exam?
Nope. Any other questions? I mean, you can ask me, well, no, you can't ask me in lab. Well, in lab, you can ask me some questions if you have specific questions about lecture material, because we'll have time in lab. So anything else? Okay, that'll do it for today.